The last video for this chapter on eugenics is actually on CRISPR. I'm including CRISPR here not as a form of alternative or therapy for gene mutation, but it is more towards of a technique or method, not only for gene mutation, but for a lot of other applications as well, to produce transgenic plants, to produce transgenic animal in various industries and whatnot. So CRISPR stands for clustered regularly interspaced short palindromic repeats. So if you guys remember your palindrome or what palindrome means, then good for you. It basically uses those kind of application. If not, then Google up what is palindrome. So the idea basically starts way back in the 1980s, but the first paper on the first paper that produced or reported a study on CRISPR is actually in 2012 by Janet et al. So in 1989, the idea starts from the description of genome editing via HR in mouse ESL by Capacci et al. So this was followed by around 3 years later by the use of CRISPR editing technology used for site-specific recombination in mice. So CRISPR is basically used more or less to cut and paste, simply said, genes to either replace or swap. In 1998, we have the discovery of zinc finger proteins, ZNF nucleases that could target specific DNA sequences, and this is basically the core of the CRISPR later on, because for CRISPR, it is important that the effective or the defective DNA sequences is being cut. So 2 years later, in 2000, bacterial CRISPR or Cas is first identified in prokaryote. And having said that, it has actually been discovered in a lot of other organisms since. In 2009, transcription-like effector nucleases or talents has been discovered from Xanthomonas bacterium. So talents basically are also a type of nucleases that is useful and being modified for the use in CRISPR. So in 2013, following the first discovery or the first paper published in 2012 by Janet et al., CRISPR or Cas genome editing, first demonstration that the CRISPR or Cas system can be used for mammalian cell genome editing. So when Janet in 2012 produced a paper, it was done in Drosophila, which is basically housefly. So this is actually done on mammalian cell. Now let's have a look at what is CRISPR, what it is about. So I'm sure you have heard of CRISPR. If you haven't, then that would be weird because CRISPR is the norm of the study nowadays. Not to be confused with CRISP, which is basically in UK it means your chips. And so CRISPR is basically based on bacterial immunity system. It functions to detect and recognize foreign DNA and later on cleave it, and of course replace it with the correct version of the sequence. So to do that, it requires Cas protein, which is CRISPR associated, that function to target and cleave sequence specific DNA. So it is more specific than your normal nucleases. So that's why you couldn't just use any nucleases, probably from some of the talents and some from the zinc finger, could be used. So cleaved DNA later becomes new spacer unit to form CRISPR array, as you will see over here, more or less similar to when you are doing or when you are going through transcription, remember? So CRISPR array, this is how you see the CRISPR array, are alternating spaces, and spaces and palindromic repeats transcribed as single mRNA. So this is how, or the basic function, or basically the basic or fundamentals of CRISPR. So from there, as you can see over here, there are varieties of CRISPR that has been basically discovered and improved over the years. So in the first diagram, or in A, you can see over here we have an array of basically repeats with a photospacer. Well, they just call it a protospacer, but it's the same thing as spacer. It's just that because protospacers have adjacent motifs, so it also have tracer RNA, which is transactivating CRISPR RNA. So tracer RNA will be the one that will guide your CRRNA or CRISPR RNA into the system, whereby another CRISPR-Cas9 complex will form, and this is the complex that will recognize the specific sequence to be cut, and it will bind to it. So once it binds to it, it will cleave the target site, so that you will only get the ones that you want. The unwanted one will be cut off. So similarly, with the second approach, using CRISPR RNA with the tracer RNA inside one whole sequences, the fusion of CRISPR RNA with the tracer RNA. So for here, you need a gRNA. gRNA is guide RNA, another RNA that is, or the function basically to make sure that it binds specifically to the correct sequence. So in the Cas9 complex, it will find the target sequence, bind to it, cleave it, and then later on, you can have the sequence that you want. Or it could also deliver the correct or the swapping of the sequences that you desire to correct a certain defect. In the third approach, as you can see over here, this is basically how they are different and how similar they are. So the differences and also the similarities between these two approaches. So you have here your CRISPR RNA, your tracer RNA, and your gRNA, how they basically function. Alright, so as you can see here, so this is the site from your CRISPR RNA. This is your tracer RNA. So it has some complementary sequences, so it will bind to it. Whatever that will be cut is somewhere here. So those will be cut. Then you will get the sequence that is needed. Probably you could just join it later and whatnot. As compared to when you use this method, your guide RNA basically is just this. And you can see that the differences between them is that over here you don't really have a 5' as compared to your tracer RNA. Your guide RNA is actually in a loop form. So it doesn't have a 5', it's basically just like this. So when it binds, the loop that you see over here, will basically stretch itself over here, and then go down here. So that is the guide RNA. Now for the core concept of CRISPR, sorry because I try to stretch this so that you could see it better, but the pixel basically becomes broken. So hopefully you can still read it. So basically a lot of times people still use guide RNA, not the tracer RNA, because guide RNA is actually much simpler and it is smaller in size. So apart from that, the first step is for the guide RNA to find the target DNA itself. So probably you don't really see how it is done in the previous slide, so hopefully you could see it better over here. So the function of guide RNA or the tracer RNA is to make sure that it finds the target DNA, because it should have the complementary sequence. Next, the Cas protein will cut the DNA strand at its supposed site, and later on it will be replaced with the DNA. So your guide RNA basically it will complex with your Cas9, and your DNA repair enzyme will be the ones that will put in the new DNA sequence and then ligate it. So this is how CRISPR is being used and utilized. So you may be wondering where is this coming from, or is there any other vehicle that brings it in? Basically it's just there in the system, kind of like similar application when you think about PCR. So you have your primer there, and you have your enzyme, and so forth, so it finds its way. So it is the same procedure. So once it has been cut, so now the replacement DNA sequence knows that it has a complementary sequence to bind to, so it will bind itself, and the DNA repair enzyme will basically seal it off. As simple as that. Alright, so in terms of Cas protein, when I do this, or when I create this slide, it was last year, there are 10 known Cas protein, but of course there are more than 11 now. So, and they have their own different function, but the ones that is being used the most is basically Cas9. So we have Cas1 up until Cas10, and there are different distributions between them. and the process whereby it requires a certain either the CRISPR RNA or the tracer RNA or guide RNA and whatnot. So and they also function differently as you can see over here. So Cas9 is the most popular and the most commonly used because it basically could be used in a large multi-domain protein, as compared to some of the others. As you can see, not too much, not in large multi-domain proteins, so quite specific in terms of the application. Alright, so the versatile nature, of the CRISPR technology is that it basically, could be used in a variety of, I would say, kind of the insertion. For example, in the first one, in A, you can see that it could be used for indel, which means that for insert and deletion. Okay? It could also be used for insertion or replacement, kind of like swapping. It could also be used for large deletion or rearrangement, if you just want to rearrange them. It could be also used, to basically activate a gene. It could also be used for other modification, such as some modification depends on what modification that you want to do and you know the stuff that you are going to do. And imaging location of genome locus, by using GFP, EGFP, which means that you could basically use fluorescent microscope to track whether it has been edited or basically inserted at the correct locus or not by electron microscope and fluorescent microscope. Alright, so these are some of the examples of editing out diseases or using CRISPR therapy. So the publication demonstrates the use of CRISPR-Cas9 for targeting diseases. So like I said, CRISPR-9 is the most commonly used among the 10 or 11 Cas protein. So in terms of cataract, so everyone knows cataract, right? So whereby you have kind of like you are not blind, but you couldn't see properly, because there is something in front of your iris. Okay, so basically what they do, is that they rescue a dominant mutation in the CRISPR gene that causes cataract. So cataract is actually caused, by gene mutation. Surprising, right? So a lot of people thought it occurs naturally at old age, but no, it's not. Okay? So you can read on the paper from Wu et al. 2013. For cystic fibrosis, so remember the disease, one of the disease that I put in as an example for gene therapy. So correction of the CFTR locus by HDR in cultured intestinal stem cells from the patient with cystic fibrosis, in 2013. So for beta thalassemia, correction of human hemoglobin beta (HBB) in induced pluripotent stem cells from thalassemia patients using CRISPR technology in 2014. We also have for HPV associated cervical cancer, to target promoter of human papilloma oncogenes to inhibit tumorigenesis so that it doesn't cause tumor production. Apart from that, hereditary tyrosinemia type 1. So you can Google out what tyrosinemia is about. So the correction of FAIR mutation in hepatocytes of mouse model of hereditary tyrosine. This would be useful because it is a hereditary disease, so it may not be passed on to the next generation once it has been corrected. And the next one we have HIV. So they are trying, so the first paper is in 2014 up until now, generation of homozygous CCR5 deletion mutation in iPSCs to propose the approach toward a functional cure of HIV-1 infection. However, this is actually not as easy as it looks like. In the model, of course, the animal model, it is fine, it seems to be okay, but when they are trying to use it using another or other mammalian cells, it's not that much of a success, or the efficiency is quite low. And the last one over here is malaria. We know that until now there is basically not really a very good vaccine for malaria production, to treat malaria. So we know that malaria is caused by Plasmodium falciparum. So this is from your chapter 2, if you remember. So they basically try to make sure that they could find some form of therapy using CRISPR to generate transgenic parasite to prevent malaria. So just to prevent another parasite to overcome the other parasite, which is the Plasmodium falciparum. And the first paper is actually in 2014. Alright, so these are also some of the experimental parameters of recent Cas9 mediated large-scale genetic screens, if you're interested into it. So they are also tested in human cell lines. And these are the comparison between traditional and modern genome editing technologies, just to cap off or summarize the eugenics. So, when you have different mutations, so this is where, if you remember last Tuesday when I asked about your opinion on whether gene mutation could be prevented or not, so we know that there are mutations. So basically, we have chemical, we have physical, we have biological, in terms of all these factors, and we also have transgenic biological factors. So these are their characteristics, okay, and these are their advantages and disadvantages and other feature. So you can see how they could be compared and be used. And as you go through this diagram, you will see that they basically are efficient to a certain extent, but it doesn't mean that they are a one type cure all. So you basically probably may need to couple it, or basically may need to use some of its advantage and to overcome the disadvantage of another mutation. So again, up until now, for a lot of diseases, there is no one cure for all. Even for cancer patient, chemotherapy actually doesn't really work to kill off all the cancer cells, because it also kill your healthy cells. So I would just leave you guys again with this. So if you have any queries, just post it under the video, or we could have a discussion on Tuesday, but probably not enough time on Tuesday. So see you next week with a new chapter on intellectual property.